Hi, everyone. I would like first to thank the organizers for this nice conference. I think we always take for granted how hard it is to organize such conference, and so a special thanks for Jonathan and Professor Evangelista and Ricardo for all all this beautiful conference. So my name is Rafael. Like Professor Evangelista said, I'm from the UTF-PR. I was going to say a few words about this university, but I heard someone else is going to say a few words in a later talk, so I'm just going to skip it. So my talk today is about helical X control in chiral liquid crystalline nanostructures, microspheres, and zigzag pattern. So I would first like to clarify this title. All it says here is actually we're just going to show some nice examples and a few of my work of how you can control and design nice applications of cholesteric liquid crystals if you have a very well-oriented or if you can orient somehow the helical X of the material. So when you see here nanostructure, it's just something that I borrowed from the materials, people that work in the materials field. They like to say things are nano very often, because it sounds better. So sometimes you get a half micron thing. It's better if instead of calling half micron, just say it's 500 nanometers. So that's pretty much what I'm going to say here. And before I go to the talk, usually people present their group in the end. But right now you're paying attention. Maybe by the end of my talk you'll be bored. I don't know. So I'm going to show some of the people of our team. These guys probably are not happy with me because I took these pictures from their Facebook profiles and WhatsApp and things like that. But anyway, so this is our team in Paraná. Of course, I'm going to present work which we did with other people as well from abroad. But this is our main group, and you know this guy, right? He's doing some propaganda of his book here. You should read it. It's very nice. We have Professor Irving. He's also here. Professor Rodolfo, he likes running. You're going to see him, his talk later. Professor Roberta, you met her on Monday. This is our postdoc, Renato. Unfortunately, he's not here. He could not come. This is a PhD student, Viridiana. She's spending some time with Professor Tony Ackley in Kent State, so she didn't come as well. And then we have another PhD student, is Renan. He's here. You probably saw his poster. Michelle, she also is here. This is Eric. He's a master student. He's hiking somewhere, so he didn't come. And this is our undergrad. It's called Julia. Okay. So, so when I first decided, I started thinking, what should I talk about when I go to this conference? I had an idea of talking about this chapter. It was published in this book. It was published like 2 months ago. So my talk is pretty much an adaptation of this chapter. So I'm going to talk a few things that are in here, and then I will jump to other things that I've done in the recent years. Okay, so if you get a chance to read this chapter, it's supposed to be good. Okay, so at least I think. Well, anyway, so I wasn't sure how much I should go into the basics of liquid crystal. So, I know some people are not from this background, and I decided to just make a quick overview. So, if you have like the thermotropic materials, the way you can go from this solid crystalline to the isotropic phase, sometimes you get like mesophases. And the most common one is called nematic. You have orientational order, no positional order is the most common phase. You have a director that is the average orientation of the molecules. But if you get a more ordered phase, which is called Ismatic A, you've seen a few talks about this before. So in this case, you have orientational order and also positional order. And this is the most common, I think, Ismatic A phase. But, there are a lot of Ismatic phases, and probably learn more from other people here. And then you have a phase, a mesophase, which is called chiral nematic or cholesteric. which is very interesting because it's just the chiral mesophase from the nematic. So people just call it chiral nematic. But the structure is very different because if you make a nematic into chiral, there's this helix, which you see here. And we usually measure it by this length, which is called pitch. Okay? The fact that this mesophase is chiral changes a lot of things. So you get a lot of optical states, a lot of things different. And usually if you want to make a cholesteric phase, all you do is you add some chiral material. Of course, it has to be compatible with the nematic phase. And that when you add some chiral material, we call it chiral dopant, it goes from nematic to cholesteric phase. Okay? So, you know, as I was saying, chirality in the nematic phase is expressed by forming a helix. So it repeats itself within a length we call pitch. And pretty much all the properties of the cholesterics are defined by this length. You know, so if you control it, you can have a lot of different things within the same phase. And usually this pitch length, it's always, the pitch length is always much longer than the molecular length. Okay, so that reflects the fact that nematic interaction is much stronger than the chiral interaction. And what's very cool here is with one single cell, you can have a lot of different optical states or different kinds of textures. So perhaps the most common one is called the planar or grain gene. You can find different names in different contexts. And pretty much what you have here, you see this uniform texture where you see the helical axis. So you have here the helix, and the helical axis is perpendicular to the substrates. And you have this monodomain structure. So it's everywhere oriented the same way. And sometimes you see those lines. These are called oily streaks. And these are just defects when, you know, for example, you're filling the material into your cell, and then you get like a spacer or something. The material has to go around it, and when it meets again, it can't just get together all these layers. If you, please. Yeah. Here I'm looking from the. This is an optical microscope. Yeah. Yeah. The pitch here is coming out, so it's like you're looking from above here. These are very thin. This is less than 1 micrometer in this case, but you have one micrometer, less than 1 micrometer here. I mean, about 1 micrometer. But you can have a thinner or thicker. It depends on the cell gap. Okay. Yeah, it's kind of the pitch is the same, but you know when it goes around and it has to, these layers have to meet again. It just forms a defect. They buckle around and forms a line defect there. Okay. This is the most common one. This is the most used one, and I'm going to say in the next slide why. But you know, if you apply like an electric field and the molecules want to reorient, you can get another. And you can break that uniform texture into a polydomain structure. People call it focal conic. I know people from this background, they don't like it. And like I always say, this is not my fault. I didn't give this name to this texture. And I think they call it because it just resembles focal conics in SMACT in some contexts. But anyway, what is happening here is you have this polydomain structure where the pitch, so the cholesterics don't want to change their pitch length. So it breaks into different domains like this. And another one that's very common is you can have this so-called Helrich texture. It's just an undulation of this. If you think about all this, the structure as a layered structure, you can see this undulation. And sometimes you see this 2D pattern where it's undulating in one direction and the perpendicular direction as well. And you look from above, you see this kind of texture. And all these textures here, they present different optical properties. The first one can reflect light. It has a photonic bandgap. The second one scatters light. And this one can diffract light if you use for like diffraction grating or something. Okay. So what is very nice here, so this is a dielectric media that repeats itself in space, and that gives sometimes this, what happens is you have a forbidden wavelength that can propagate through, so it just propagates, it cannot propagate through the material. This is called a photonic bandgap, and this obeys what's called a Bragg reflection. So if you shine light in the same direction as the pitch length or the helical axis, what happens is sometimes you depending on the pitch length, you get a reflection of some wavelength interval. So, you know, if you measure this, you often get a reflection by wavelength. You can get this nice peak here. So it reflects circularly polarized light in the same sense as the rotation of the cell. So this photonic bandgap is defined by the ordinary refractive index on the left side times the pitch, and the other side is the extraordinary refractive index times the pitch. So, you know, if you look here, if the center is around 600, which it should be here, you get a pitch length with about 400 nanometers. Okay. So this is actually one cell or a set of cells. So you can make like blue reflecting cholesterics or green or orange light. Okay. So okay, let's talk about the helical X. So definition of helical X is very simple. It's the x of rotation along which the director rotates. So director is a numeric, like I said. It rotates around in space, and you can define a helical X, which I show here as a red arrow. And if in most of the cases I'm going to say here, we're going to talk about strongly twisted materials, which means the pitch length is much smaller than the cell you are using. So we call it strongly twisted. And in this situation, you can consider it as a lamellar behavior, which means the material doesn't, it costs energy to change the pitch. So instead, you can just think of kind of a symmetric-like behavior where you can just change this helical X direction. And I'm going to show here that makes a big difference if you can orient this somehow for several applications. And you know, people usually use coarse graining models to describe such behaviors. You can see this paper here as one example. Okay, so what difference does it make if it's oriented or not? So one example here, this texture is called the fingerprint texture. So of course it resembles a fingerprint. So here you just take like a homeotropic cell, you put a material there, and you see this kind of texture. And of course, you can see here, it still maintains the pitch length, but you cannot find any orientation of these layers like you can define a helical sense here. On the other hand, you can put this exactly same material into a hybrid. What you see here is you can define this layered structure with a very well-defined direction here. So the first case, which is the fingerprinted texture, you can't really find any application for it. But if you use like in a hybrid cell, I'm going to explain in a few moments what is going on here. You can have several applications of this structure. For example, for diffraction gratings. Okay. Anyway, so what happens when you put a cholesteric in a hybrid cell? So hybrid cell means you have one substrate that it's a strong tries to orient the material in the planar direction, and the top one that tries to make it homeotropic. And this caused some frustration. I think people have discussed this a little bit here before. And you get this undulated, so this is a simulation, the results. You get this undulation where you have kind of a perpendicular direction here of the helical axis. And you see below here you have some space which where the material is still planar. So what happens here, if you try to change the pitch, this elastic boundary here, it tries to rotate, and as you change the pitch, the whole thing has to rotate. And if you see from the side view, it should look like this, and when you look from the top, it looks just like this. Okay. No. Here we have those. I mean, there are defects, but there are cholesteric defects, like no core, those lambda lines. Yeah, but no core lines. Maybe it depends here, maybe on the substrate. Yeah, but no, in this simulation we have weak anchoring here. That's why it looks like this. If it's stronger, the whole thing is pushed down to the bulk. And in this case, I don't remember if it has a defect here or not. So why control the helical X? So you can, like I said, you can define very nice applications. Usually, in some cases, you need 2D or even 3D control. And what I'm going to show here is you actually have several ways of controlling the helical X orientation. You can apply field. You can shine light. You can use confinement, and there are several other ways of doing this. Before I go to my work, I'm going to show some very interesting examples here. I'm not sure if you guys are aware of this paper. It was published in 2006 by the Faringa group. It's a Nature. It has just one page, and the last time I checked, it has over 600 citations. And it's very simple. All he did here is he took an emetic sample, doped it with a chiral dopant, and this chiral dopant, when you shine light, the molecule changes the conformation. And as it changes conformation, the helix has to either stretch a little bit or compress a little bit. In a better sense, what happens is you change the helical twisting power of this material just by shining light. So when you put in a hybrid cell, like I said, you form stripes, right? And here you have a sequence of photos where he shines UV light, and the pitch starts to increase. And the only way you can put less layers here is by rotating and nucleating, moving defects around. Or like I said before, you have that little elastic layer there. And what is interesting here, he places a spacer here, and he observes the rotation of this spacer. So they claim, of course, you have a molecule, and it can move these objects that are 10,000 times larger. For this reason, they call it molecular motors, and for this reason, Faringa got a Nobel Prize in 2016. I'm not saying about this paper only, but for all his collection of papers. And other examples, you can have, like this one, you put a cholesteric, and then you watch a wetting transition, and after wetting transition, you get a very thin layer, and if cholesterics are in contact with the isotropics, you have to orient it somehow, and what happens is you form this very well aligned stripes here, and those stripes have a well-defined helical axis, and just by changing temperature, you can control the thickness of this wet layer, and you can observe rotation and all this movement, and this is controllable. You can go back and forth. Other applications, you can see, like for display applications, I'm not sure if you're aware, but you can use cholesterics for displays because it reflects light. So you can make a display that doesn't have a backlight, right? So it should be great for our displays because you can save a lot of energy. But the main problem is the cholesterics have a very slow response time when you apply a field, and then you destroy the helix. It goes to homeotropic orientation. The time to go from the homeotropic back to the planar state takes a very long time. But sometimes you can just apply a very fast pulse. You have to design a way of doing this. You can find some information here. But what happens if you apply a very strong pulse, you can get this wrinkled state, and it turns out this reflects light, and this just reflects light, but this light eventually doesn't go back to your eye. And here is when you see under the microscope, and this is a very easy and nice way of controlling. This response time here is about 2 or 3 milliseconds. So instead of using 300 milliseconds like the ordinary way, you can make a simpler cell design like this one. Okay. You just apply a very short pulse because what happens here is when you apply electric field in the cholesteric, so the molecules try to orient. If it's positive dielectric anisotropy, it tries to orient with the field, right? So what happens is it has to unwind the helix, and what happens is this is a nucleation process when it happens, because first the helix tries to go to focal conic and then to homeotropic, and this happens by nucleating defects around the spacers and things like that. So if you apply a very fast field, very fast pulse, there's no time to form this focal conic state. What happens is you just the material just try to get a wrinkle configuration like this, just to get rid of the elastic energy. So we apply like a 1 millisecond pulse, and then it forms this wrinkled state, and it stops reflecting light. So, well, it's kind of this. Here we're just looking from the side, right? So if you think this is rotating over here, it tries to form a conic, but you know, such materials don't form cone states very easily. You know, like TB, now people are finding this. This is another example. This paper was published by Kwon Lee at Kent State University in 2016. And here they have a cholesteric. It's made by doping a chiral dopant that responds to light. And all they do here is the cell is made to induce planar alignment. And then when they shine light, their motor, their switch they use here, the chiral dopant, is very, it changes configuration very abruptly, very fast. So you don't have a uniform, if you don't illuminate the whole cell, you get all these very states here because you have different cells that they have different pitch. And essentially, you can control the orientation of the helical axis in a 2D form, and eventually you get back the planar state, and then you can also induce the helix in the other sense. So only by shining light, you can control the helical axis in a 3D sense. You can control in the plane, and you can control from the top to the bottom. So you have the rotation of the helical axis in a 3D manner, and it's very controllable. And the only thing, only stimuli you need here, only stimulus is example. Here people use it, you know, usually you have buffed surface, so you have to rub the surface, and that causes a lot of defects, and that's not very good for precise alignment. So in this paper here, people use it, an alignment layer that is, you use some molecule that is, some material that is, it reacts to light. It's called SD1. It's an azobenzene material. And if you shine polarized UV light, you can change the orientation in the on the substrates. So they can design several different patterns. They put a cholesteric here, and you have this radial orientation of the helical X around certain directions, and you get like this very nice pattern. This looks like a Fresnel lens, if you will, although it should be a little larger here. And in the paper, they show a lot of different ways of doing this. Okay, so I'm going to jump to things that I've done now. So one very nice way of the helical X orientation is by, instead of putting the cell into like a slab cell, you just make into a sphere. And if you make those droplets, you can actually have a radial distribution of the helical X. And it seems like people have been paying attention a lot about these droplets now in the past few years. This has always been a curiosity, but in the, maybe in the last 3 or 4 years, people are paying more attention to this. And this is one example of a cholesteric droplet. So here the pitch is long, so you can actually see the stripes, right? This is a paper published by Zoomer Group, where they run simulations and explain a lot of the orientation of the director inside of these droplets. This is a planar-oriented droplet, and what happens here, you know, you have this kind of double twist orientation here, and you get like a point defect at the surface as a Bojun. And people thought before you would have like a defect lines here, and they just showed in this paper all you have is just non-singular lines here running here. But there's still some debate about this. But anyway, it's sure. Yes, yes, but not a singular line. That's what I'm. Yes, yeah, sure. I'm saying about people are usually with use it to lines in nematics, so that's why I'm comparing to those. Yes. Yes, sure. Yeah. Yeah, yeah. You can definitely see. Yeah. Okay. Thank you. In my applications here, we want to make those droplets with very small pitch, and we want to control how it reflects light. And here you can actually get not a 1D photonic bandgap, but a 3D. Okay, so you know it's important to do those things. You have a lot of possible applications here, like communication systems, sensors, and biosensors. People have been using these droplets for this goal and for cloaking and camouflage. And you know, you can search the literature. You're gonna see a lot of those applications for these droplets. So what we did here, we use this photoisomerization. This is one example. So this is a chiral dopant. This molecule is chiral. You put this in a nematic. You see here there's an isobenzene group here. So you shine UV. This thing goes from trans to cis. And when you do this, so the green bars here, they represent like the chiral dopant. So it changed the shape of the molecule. And this has to stretch the whole helix. So before illumination, it looks like this. After, it looks like this, so it's longer. If you measure the reflection spectra, what you see is after 10 seconds, it goes all the way from blue, reflecting all the way to like infrared. Okay, and you can see all the states by looking here. So we did this micro fabrication of the droplets. This is a standard procedure. These actually were made in David Waite's lab. So we send him his group the material, and you have pretty much here two fluids that are flowing inside of this capillary. One is the liquid crystal mixture, which is composed of a nematic doped with a chiral dopant that is photosensitive to light, and the other fluid we call the outer fluid is just water with PVA. Okay, and there are two reasons why we use PVA. One is so the molecules in the droplet, they will anchor planar. People have known this for a long time, that PVA induces planar anchoring, and also because PVA helps to stabilize these droplets. Okay, so usually 10% of PVA. Here's you have this whole thing flowing, and you get you know, by these speeds you can control the size of the droplets. And in this case here, we fabricated the 50 micron diameter droplets. So we use this material. This is a more stable one compared to the azobenzene. So people from the chemistry department, for the chemistry group in Kent State, Kwang Lee, they synthesized this material. It's very nice because this is thermostat stable. It is a open closed ring, and you know, you shine green light, it goes from short pitch to longer pitch, and if you shine UV, it's reversible. So we fabricated those droplets using this procedure. So here is a transmission light when you see under the microscope, and this is what we believe it's the helical X organization inside the droplet. Okay, so we have this radial distribution here. And what's interesting here is how light interacts with those droplets. So, like I said in the beginning of the talk, these droplets, they obey like a Bragg reflection criteria, right? So, if you make the pitch, you can tune it to be reflect red light. So, light incident when it goes directly above the droplet, you should see a spot that is red, right? But what happens as you go far away from the center of the droplet, the helical axis starts to tilt, right? so you should see different colors. And what happens usually is when you go like in the middle of the droplet, it should reflect because of this criteria. So cosine theta, you should start seeing green light, right? But usually this light just is total internal reflected here, or the microscope doesn't collect it. But what is more interesting is when theta gets to 45 degrees, so this is a basic reflection rule, geometrical optics, you get these incident lights reflect with the same angle, so it's also reflect with 45 degrees, right? So what happens is light goes and finds another droplet, and then it's reflected back up, also with 45 degrees. And if you make a cholesteric that reflects light, this light that reflects to the other droplet should be blue. Okay, so what you see is a kind of cross communication between the droplets, which is in another wavelength, not the one you designed from the beginning with. And this is showing it a little bit better how it happens. So you have you shine with white light, red lights reflect your eyes because of the band gap I told you before. But some light it makes a 45 degrees angle with the helical X is directly reflected to the other droplet, the neighbor one, and that one is reflected up again. So you should see blue light communication between the droplets. And this is what we should see from above. So when you do this and you put all these droplets together, you see this very nice dispersed array of droplets. You actually see it. So we tuned the pitch to be red in the center, and you see this very nice spot here. There's red. And if you pay attention, all these blue rays, which look like arrows, they are pointing to neighbor droplets. So, for example, here, you don't have a droplet here. There's no blue lines here. And this is very nice. This work was published in 2015. Here's how the Crosstalk works. So if you shine just in one droplet, you just don't see any communication. You just see red light, right? But then if you put a neighbor one, you're just going to see they communicate. It's kind of, you shine one droplet, it tells where the other friends are, okay? And you can design all these very nice patterns. They have this cross communication. And because we have a light-sensitive chirodopant, you can play with the way they reflect light. So here we have initially reflecting blue. So here we change the gain so we don't see this communication. We put a low gain in the camera so we don't see communication. And you can tune the pitch so it's reflecting blue, reflecting green, reflecting red. And we can control single droplets, the reflection, right? So we can make like this one red, this one green, this one blue. And you can design patterns like flowers and things like that. So. And other interesting things, we can design several patterns. A very ingenious way of doing this, you just take a very thick cell, you put those droplets, you put the cell like you know, not straight, but in an angle, and all the droplets go to the bottom of the cell, and you form this double layer of droplets, and you can make these very nice patterns. You can control the color the top, on the bottom, and things like that. So after this, we made another set of samples. In this case here, we made samples that we use a different chirodopant. And what we did is we used this chirodopant and a regular one that doesn't suffer isomerization under when you shine light. Okay. And what happens here is before illumination, the left-handed chirodopant, which is this one, right? It dominates because the other one is right-handed. So the material is left-handed. But then we shine UV light. This one is weakened, and then it goes to right-handed. So you have a droplet that is cholesteric. You shine light, it goes to nematic, and then goes back to cholesteric with a different handedness. And we can see the dynamics of this happening. So this cell was made on purpose so we can have a long pitch. We can see the lines here. And we shine light. We can see this very interesting dynamics here because it starts to unwind, and the droplet goes to a very tight, many turns here. it goes, the pitch decrease, eventually becomes animatic, and then it starts winding to the other direction. And eventually you get different rotations to the other direction. This paper was just published end of last year. What is very interesting here is if you think of what happens to the defects here, and what we think happens here, we have these non-singular lines I mentioned before, one Beaujon. Eventually you get fewer turns, and the droplet adopts what's called a twist bipolar. So these Beaujons, they start to move around until eventually form the bipolar structure that people know with two. Go ahead. Yeah. This picture is not that great. That thing still exists here, but it depends how you can. That thing can be on the bottom, and sometimes you don't see it. So actually, there is some debate. It has to have many turns here to form that structure, and the people are discussing this. So you can have these different structures in the same single droplet. We're currently doing some simulations about this. We're trying to understand how these defects move around. We're doing Landau-Digenis simulations. People have shown this before here. So we have a bulk energy, surface energy, and we add here dynamics. This is pretty much the same modeling Zoomer does in his papers, but we add here the dynamics because we want to see how it dynamically changes. And right now we're getting some interesting results. Nothing is published here. Is read. I apologize for this droplet. I realized I put this here yesterday, and I realized I didn't have this initial configuration on my laptop. I have on my desktop computer, but the CSV data is too big. I could not download here. So this is not exactly this configuration. This was a little bit before it relaxed to this double spiral. But you can see as we change the pitch, eventually same thing happens, and you get this all these transitions, and we can actually see it happens. This initial configuration is just like Zoomer predicted, and then it goes to this twisted bipolar configuration, and eventually goes to the regular bipolar configuration. And you know, we expected that when you reverse again, if it's too fast, you can get another other metastable states, but we're still looking for this. So we also made samples that were short pitch, and if they are short pitch, you have reflection of light, so you have blue light communication. But if you make the pitch longer, eventually here is like orange, then it becomes red here. So the cross communication should be green instead of blue, and eventually you don't see any reflection here. So the pitch is too long to reflect light, but you still have reflection and cross communication. And you can go all the way, passing through nematic and then winding back again until you go to reflective state. So this is a different different amount of chirodopamine. Okay. Okay. So maybe I'm running out of time. I'm going to show a different application here, which is we also made this zigzag shaped. So it's very interesting to often study patterns, and sometimes you just need to use like a synthetic material, like a cholesteric, to design different patterns. And if you look around in nature, you find a lot of these zigzag patterns, like in leaves or spider webs or even graphene, and all have different properties. So if you look in liquid crystals, there are some reports of zigzag patterns, like in smectics, ferroelectric smectics, or nematic over sinusoidal grooves, and you can look this up. But none of those are controllable, switchable, or have any application. So we design. This is the planar structure. We design a material that is originally planar. I'm going to show more details later. But anyway, if you have, like I said before, if you have the fingerprint, you don't have any application, right? But if you have this line helix structure, you can use for many things, okay? And what people usually don't, if you don't work with cholesterics, you don't know, but these stripes, they can have different families. They're not always the same. actually you can have this developable modulation, which is like you apply a field in a planar cell, cholesteric, and you form stripes, and they just appear everywhere, like a second order transition, if you will. Or you can have a growing modulation, which is just like a first order transition, so the stripes grow slowly. You know, in here, one paper, you can look it up, you can see these examples. So what has this been used for is for like a diffraction gratings, and if you can control this, you can have a switchable grating. You can change the diffracted angle. You can you can change the diffraction vector. A planar cell, 3.7 micrometer, and this material it has helical inversion. You can observe this. So if you put in a planar cell and then you apply electric field, you see this stripe pattern, right? And then we start shining UV light. The pitch has to decrease. So eventually the stripes disappear. You go to a sort of a conical structure. Eventually you get an unwinding. It goes to homeotropic state because you have a 1 volt applied here. And then eventually forms again when it goes to the left-handed configuration. So what happens if we keep shining? Eventually we start seeing like a zigzag pattern. Okay, so maybe this picture is not that good. We had to use like UV filter here for the microscope, so I'm going to show better pictures later. But all that happens here is you have the straight lines, and you keep shining light, you get the zigzag patterns. And you can see it here, more or less. You keep shining light, the zigzag angle increases until eventually the pitch gets too small for that field to hold the stripe pattern, and eventually goes back to planar. So what's going on here? So these pictures are better. Here you can see the zigzag pattern. So we start, if you look at this blue line, you have D over P. This is the thickness to pitch ratio. You apply UV light. It goes from 1, eventually goes to nematic, and it keeps increasing in modulus. And in this region here, you get a zigzag pattern. If you look papers around, people say that in this area, usually the lines, the stripes, they can form either perpendicular to the rubbing direction or parallel. So when you make a regular cholesteric, you can get either of those. But what happens when you transit from one state to the other? And you know, in order to investigate this, we use this simulation process. It's called the Q tensor. Is to Landau the genes, we use the representation of the Q tensor, but instead of using the Landau energy, we just use Frank free energy. Just I think people have shown it before. I'm not sure if it, in your case, was the tensor, but maybe similar. So we do the simulation, we go through all this process. So we keep changing the D over P. So eventually, with that field, you have a homeotropic configuration. Eventually, you get this sinusoidal configuration of the director. And as the D over P increases, there's a lot of energy. So here, these colors represent the elastic energy at the surface, which indicates the stripes want to change direction. And what you see is you start seeing this zigzag pattern happening. This has been published last year, in case you want to look it. And you get this very nice controllable zigzag pattern. You can turn it on, turn it off. You can control the angle, and we can use this for controlling diffraction of light. So you can see here you get this. You can change the diffracted angle. You can change the pattern. When it forms the zigzag, you get these brackets, which you know if you're not very straight lines, you can start getting. You want to ask a question? Initially, so here, I mean, if it's planar, yeah, it's planar cell. No, it's planar cell. But when you we use an electric field and you shine light at the same time. So it forms stripes, and those stripes form the zigzag. So you get all this nice control of the diffraction patterns. If I had more time, I would explain a little better. So a few conclusions, just we can make these very nice droplets, control the photonic properties of them, form these different patterns, cross communication. We can design zigzag patterns, we can control it, we can start, we can terminate, and you know, we have some explanation for the results. We can use these as many different applications. So I would like to thank the organizers again, my university, CNPq, of course our ENCT, so Professor Figueiredo is here, and okay, thank you.